Okay, so this is about the cell cortex, the physical properties of cell cortex. So we already know from, particularly from Guillaume's talk and from Vijay's talk, that cell cortex is a very, very important candidate in the environmental development of any organism because it's flow and forces together. And the flow and force is very important to have a particular shape of any organisms. And this flow and force is in turn is governed by the material properties of the cortex, right? Because it depends how it flows, it depends on the viscosity. For in case any material, like the flow of water, flow of honey, is depends on their viscosity itself, for example. Right? So here, what we are going to do that we try to estimate the material properties of the cell cortex that eventually control the flow and force of the cell cortex. It will influence the shape of the organisms. So this is very common example of embryonic or morphogenetic processes going on during the developmental of episodes of a organism. So this is this one is zebrafish epibole. I will go detail into that, and both are the same and from different viewpoint. So this one is the enveloping layer, which Guillaume has already talked about, and this one is again the full thing is developing. So, so this is a paper by us from Guillaume and others from CP's lab that what they have. So, so this is the full the epibole of the organisms. So here we see this is the enveloping layer, the multicellular layer, and then there's a bright ring formed by actomyosin, which actually contracts, and the mechanism of the full epibole process is well explained in Guillaume's work. So there are two mechanisms which this actomyosin cortex, there is contractility which can couple to the curvature and to the friction. So they have shown that the friction is more important than curvature for this contraction. And then the second important model system is C. elegans, where this one is an eggshell and the full worm is developed within the eggshell. And there is also lots of flow that we have seen. We are seeing that this is more. So Vijay has talked about more in detail that what are the flows, or what are the what these flows doing. So I'm not going into the details. So he has already mentioned that this kind of size asymmetry is there in the first cell stage division. And there is also compositional asymmetry in the same process. So this asymmetry actually decides what the fate of these cells in the future stages of the development. So Vijay has been talked a lot on this. Now, so the point is here that. All the flow or the things that we are seeing here that actually affects very much about the whole developmental developmental stages of any embryonic development. So this is kind of we can say from the more physical point of view, any developmental process is a intertwined dynamics and that is why the cortex is becoming very important candidate, because it can create force centers by, for example, myosin foci, and this force can trigger flows, and then the flow in turn can reorganize the force centers, so the force profiles become changed. And so this is kind of a coupled system of force and flows. And also a very important point was there, as already discussed by others, that there is a signaling. So from when the force will trigger or when the flow will start, that is coming from some signaling process. So the whole developmental process of any embryo is a coupled process of signal, force, and flow. And my job is here to convince you that all these force, flow, etc. are dependent on the material properties itself. And that material properties, some of them to some extent, we will estimate or we will calculate it by a particular process. I'm going to explain in what process we can estimate those material properties like viscosity, like active contractility or friction. Okay, we will see that. So I guess the introduction or what I'm going to do is more or less clear, particularly for the students here. So now, another thing is already mentioned by other speaker that this is the flow that I'll be concerned here. One of the flows actually, I'll be concerned two different kind of flows. So one of them is this is a C. elegans. So this is the first cell stage division, and the flow from the posterior end to the anterior end. And why this flow happens? Because of the force imbalance or the stress imbalance. And from where this stress is generated, again and again, I guess mentioned other speaker, that is the source of is this. So there is myosin, there is actin. So myosin works and uses this ATP, and they generate the force, and there is an imbalance between the traces. So in this paper, they have explained why this flow is there. So this flow is because of there is differential stresses from this part and this part, and they prove it by laser ablation that the stresses are indeed different from this posterior part and the anterior part, and this difference causes the flow. And then there is again that I have talked about earlier. This is the same mechanism that follows in case of zebrafish as well. So there is actomycin ring, and that contracts coupled with the friction. So friction is coming from the cytosolic layer here, and the retrograde flow that happens from the vegetal end to the animal. So the vegetal pole to the animal pole. There is a continuous flow of myosin, and there is microtubule as well. So these are the cause of the friction that felt by this ring. So this contraction coupled with the friction and also the curvature that actually these are the players of this contraction and the full epiboly processes in the macroscopic scale. In microscopic scale, of course, there is ATP and that hydrolysis to generate energy. Now we are trying to estimate the physical properties of this actomycin ring. So again, that I have already shown. The flow depends on the material itself. That is why honey is not like as fluid as water is. So it is necessary to estimate the relevant material properties of the concerned fluid to understand the flow itself. And for that, we need to perturb a fluid. So this is the same philosophy where we measure the viscosity of, say, water, like using Stokes kind of experiment. So we perturb the water using the still ball. So here it is more complex fluid. So we are using the laser ablation, and it's more sophisticated animal there. So the thing is we have to perturb the system to measure something. That the perturbation method is laser ablation. And here it is. So this is the example from Epiboly, and this is a lab from CP that they cut the. So this is the actomycin ring that form at the stage of 60% of Epi, 60 to 65% Epiboly stage of the zebrafish, and here this is actomycin ring, and we cut a laser. So the goal is, so when we cut a laser, due to activity, the contractility, there is a flow surrounding the laser cut, and we will analyze the flow in some way, and from that analysis, our physical parameters will come out. So first step is the analysis of the flow due to the laser cut. here. So same thing that has been done in Stefan's lab is this, that there is this C. elegans flow, and then they cut. So we can cut along the axis or perpendicular to the axis both ways or any direction. So depending, so whether the underlying network of actomyosin is isotropic or anisotropic, that will have an impact on the flow surrounding the laser cut. So these are the examples that the people do in the lab while doing the laser cut. So we will be interested to analyze the flow surrounding the cut to estimate the material parameter. Okay. So so first step is clear. That first step is the analysis of the flow. So how you do it? So this is the first the first point of the analysis. That's the intensity analysis. So where we cut here along the line, immediately after the cut, the things becomes dark because there is no actomyosin, and this is all myosin is labeled. So we see a region. Then the turnover of myosin and actin both happens, and then the recovery. Actually, this whole process is called the recovery. The network recovers itself. So there is a time scale involved with this recovery, and we can see a nice curve of the intensity versus time, and we assume that intensity is proportional to the myosin density. So it is where it is more intense, there is more myosin, and less intense, less. So this is so here we can have a idea where the myosins and how the myosin can turn over throughout the time after the cut. This is our first quantification. Second is the velocity. So first is the density of myosin, and second is the velocity perpendicular to the cut. So this is the PIV analysis that Tamal mentioned the basic physics of PIV, that we take two adjacent frame, which are here it is like differed by 0.5 second, and then we do the intensity correlation, and from there we can get this velocity profile. Interestingly enough, that this flow. So here we have this cut. From there, this velocity profile decays. So we can get a certain kind of length scale. So the previous analysis with intensity gives us certain kind of time scale of intensity regeneration or the recovery, and here we can guess some sort of a length scale from the velocity decay from the away from the cut. Okay. Then there is a shape. So when we cut on a 2D, say for example a rubber sheet, then we create a hole, and if it is stressed, then it is kind of a ellipse. So here we have this ellipse due to the cut on the actomyosin layer, and we can analyze the shape following way. So we can fit an ellipse, and then we can plot the half of the minor axis of that ellipse, and there also we can get a time scale. So note. So color coding is again intensity. There is a contour plot. So if this is the red one, the more myosin intensity, and so here, so this is the point. So though it seems that from the color code and also that there are myosin undulations or the inhomogeneous or the heterogeneous thing. So in our purpose, later we will see that we assume a homogeneous distribution of myosin, which is not a purely true thing from the experiment itself, but it can estimate quite a good amount that what is the parameters are. But the theory is developed in some way that we can incorporate the inhomogeneous nature of the myosin itself, but it is not yet done. So this is. Okay, so the point is here we get another time scale, right? So this is roughly of the order of 5 seconds. The time scale of the recovery is roughly of the order of 30 seconds. So there is a second time scale. So we can at least have an idea at this point of time, without doing any theory, anything, that we, in this problem, in this flow, due to the laser ablation, we have two time scales. One is this opening, right? And the other is the recovery, right? And one length scale. But these are all just from the experimental data analysis, nothing else. So no accurate number I can present you at this point of time without any theory. Okay. So this is that the whole thing, just to have a summary, that we have cut in both this is C. elegans, this is zebrafish cartoons, and we cut and we see the flows here. And similarly a similar thing, we also, so the length scales are different. The zebrafish embryo is big enough. It is like 350 micrometer in the radius or something like that. And this is much smaller, is a 50 micron kind of the one cell stage. But still, the phenomena-wise, are quite similar, because everywhere the actomyosin is involved, we cut the laser with the same power and all, but the scales are different. So these are the analysis of the flow, fine enough, but then what about the material properties? So how to extract the numbers out of it? That is our major thing now. So for that, we really need a theory which couples the flow variables to the material property, right? So at this point, I thought that better is a little caution about this thing, that there are many certainly different kind of theories, coarse-grained theories, microscopic theories, and Guillaume also presented in the tissue scale, there is the vertex models and all. So, but it is very difficult, as the system is very complex, it is very difficult to find a universal kind of theory. So anyway, we will go with the active hydrodynamics. So at this point, I thought that maybe as experimentalists are also there, we should have a glimpse that on which this active hydrodynamics works. So we'll go the outline of the theory that we are going to use, in particular the students here. So before going into that, we kind of crudely classify the active matter system. So one is the length scale that we have, this viscosity over friction, length square actually. And if it is very small, we say it is dry active matter, which might not be very important for the biological or the morphogenetic things. So it's more like flocking and all. Then if it is very large, that is more viscous, more fluid, we have this wet active matter, which is more important in biology on this morphogenesis context. Then within this, so we are here to analyze, and then there is a time scale separation as well. So we have a relaxation, various relaxation time scales in stress relaxation time scales and all. For very small time scales, we can have a viscous fluid, viscous active fluid, and if tau is quite large enough, then we can have this viscoelastic kind of behavior that Pramod just talked about the neuron scale. So a crude kind of classification of active matter, and then we go a kind of formulation. This is particularly important for the students here, not like for heavy users of this theory like Guillaume and Vijay and others. So this is like the entropy. We write the entropy production. So the generic scheme is like writing entropy production in terms of a thermodynamic force and flux with a bilinear form. That is the product form. So here, for example, mu and del rho del T are the thermodynamic force and flux, like this. So this is the entropy production rate, and then it is coupled with the momentum conservation and the mass conservation principles, and we rewrite the entropy and this entropy production regarding this. Then we identify what are the flux and force. Of course, we know the flux and force, but we identify their signatures, like what are they, with time, what are the forces that change the sign of it? Example, velocity, right? So that changes the sign with time, right? If t going to minus t, then it changes the sign. So these are the signature of what are the reactive or dissipative terms of the of this entropy production rate. So then what we do, the next step is we expand the flux like with linear, assuming the linear Onsager kind of expansion, and then we respect the symmetries of the system, of course. What are the symmetries? Like the spatial, this one symmetry, that whatever we expand, we have to consider the tensorial character of the, like for example, stress, the density is a scalar, so it is a rank 0 tensor. Then the velocity is a vector, and then it's the stress, which is a rank 2 object. So this tensorial character we have to keep intact in the expansion, and then we have to respect the time reversal symmetry as well, where the dissipative flux and reactive flux will come. So with this all, this is the relevant reference for that. For the students, I would say that just go through it and have a feel what is the theory involved inside. Then you can see that. So with this approach, we finally get a list of dissipative flux, that the flux and forces, and that and a list of reactive flux. With this, actually, these are these will help us to get a constitutive relation of the fluid that we are working. Okay, so for our case, it is much simpler. We don't have polarity, we don't have pneumaticity, all those order parameters. We have only density and velocity and stress. So the constitutive equation of the fluid that comes from the theory is much simpler. So we will, okay, now I go term by term. This sigma alpha beta, that is the stress, total stress involved in the actomyosin network. Then this is the active stress. So the whole, this thing becomes a passive thing, right? So inside that, it is involved that the total stress is a superposition or a sum of passive and active part. Okay, so this is the whole thing is the passive part. Sigma alpha beta is the total stress, and this is the active stress. Now, what is these terms? So here we assume the active stress is isotropic, right? Therefore, this delta alpha beta comes, and this is the contractility factor. So the chemical potential that is used to hydrolyze the ATP to ATP and create the energy. So this is the total thing. Now, say for example, so first consider this one into this, okay? Forget term. So this one is equal to this. So this means that this is the shear viscosity, this includes the bulk viscosity, and we have this strain rates, right? So this gives this says that the stress is proportional to the strain rate, and the proportionality constants comes as the viscosities, which are the material parameters that we are going to extract. Then what about this term? So this comes from the elastic factor. So this fluid is not fully viscous, it is viscoelastic in a certain manner that for small time scale it will show. This is very important, like within the time scale, this is a time scale, so within that time scale it is elastic, beyond that time scale it is viscous. So it is a kind of Maxwell model, the Maxwell, this is a physical model people say. And so this is, we consider here through the constitutive equation, it is an active viscoelastic fluid. Okay, and also one very important point, we consider it is as a compressible fluid, right? So because we have this divergence of velocity, we consider to be non-zero. Why it is compressible? Because we consider the turnover mechanism. So the density can change, so the actin and myosin both can turn over, and we say this is a compressible fluid. That is why two different kind of viscosities are there. One is shear and one is the bulk, and they are related. So they are not independent parameters. So this much about the viscoelastic active compressible fluid constitutive equation. Then there is a force balance. Again, a paper from Guillaume in 2009. Says that the stress, the gradient of the stress is proportional to the velocity itself with the proportionality constant gamma. Okay? and this proportionality constant is the friction. So we, from here, together with the constitutive equation, we can set up a length scale that came earlier into the classification of the active matters, and we have this length scale now. Together with these two equations, we have now the equation, the dynamical equation for the density of the myosin or the regulator. So we assume that myosin can diffuse throughout the network. So this is the diffusion term, and this is the advection. So the myosin can transport it by the flow of throughout the network. So this is the advection term, and this is the turnover, right? So all this, so these are the model equations. So these are the theory part of this thing. So we have, so the physical principles that go in the theory is like, so the stress is proportional to the viscous strain rate, then stress is also proportional to the elastic strain. That is how we are considering the viscoelastic constitutive equation. Then the total strain is a sum of viscous and elastic, because you know this Maxwell model has this series connection of damper and elastic spring. So this is here, and this total stress is a sum of passive and active stress. And this is the rate, this is the height, it should not be the height, but anyway, this is the density profile change that I show you in the last slide, that here. And this is the diffusion, advection, and the turnover part. And we also consider the cortex to be a thin film, and the gradient of stress is proportional to the velocity that gives the force balance. So here, so the material, this one, parameters involved in this model, in this theory, is like these two viscosities, then the relaxation time scale tau, the viscoelastic or the elastic time scales, then there's the turnover time scale and the friction coefficient, then the active contractility coming from the active stress. Okay? And there is the flow variables which are involved in the equations. So these are the, so now we are ready with the theory, and we have already analyzed the experimental data. Now it is the theory. And first check what the theory can do without any comparison with the experimental data, say. So this is what a theory can do. So we can set initial and the boundary condition as the laser cut. So where we cut the laser, where we cut the network through the laser, we assume that the stresses and velocities, etc., density as well, immediately goes to 0 along the cut. And that, and then so now we can calculate the response of this cut. So this profile is the density profile, and the arrows are the velocity that we can measure in the experiment. Okay, and these are the different. So this is also the distance, the velocity profile that decays from the cut that we also get in the experiment. So these are the things that we can calculate from the theory, and we can compare with the experiment. So the rest part of my work will experiment, but this is just showing you what theory can give to you without a comparison. So now we are going to estimate the material parameters, and we will follow a flow, like a flow chart, like what are the processes. So first we find the independent variable or independent parameters of the theory. So that will be like non-dimensionalizing the equations. We will get the independent parameters. Then we solve the model equation with laser ablation condition, like along the line, all the stresses, etc., the density will be 0. And then we start to see the evolution of that. And then we get minimized, like, so we calculate the similar thing that we get from the experiment and we get from the theory, and we minimize the distance between them, like we try to compare the experimental and the physical and the theoretical part, and we try to minimize the difference between the experimental and the theoretical data. From there, we can extract the material parameter of the actomyosin cortex. So what are the parameters, independent parameters that we can calculate from the theory? So here we know very, very precisely the lambda c, that is the cut length of the, and then we have this lambda, another length scale that comes from the flow itself, which is the friction over, the viscosity over friction of this under root of it. So this we can calculate because we know alpha c, lambda c, this cut length very well. And then we can also calculate the stress relaxation time scale, the elastic stress relaxation time scale, because from the independent experiment we can know the tau a, that is the turnover time scale. Okay? And we can calculate active contractility over friction. So these three parameters that controls heavily to the flow, the active flow, due to the laser cut, we can extract. Okay. Cut length is a kind of given. So when you cut a laser, cut actomyosin length, you have to fix the cut length in the laser ablating the machinery as well. So from there you can, so you know the lambda c as well. No, it matters to the, of course it matters. The thing is, you know the cut length, but where you will place, like perpendicular, for example here in case of zebrafish, whether it will be perpendicular to the EVL or the parallel to the EVL, that matters to, so the parameters that we will extract here will differ slightly whether you analyze the perpendicular or the parallel, due to the anisotropy of the active tension lying at. So here we will, I will present the data only for the parallel cuts, but same process follow for the perpendicular cuts as well. In case of C. elegans, it's also fine. Like perpendicular and parallels are different. That is why the flow induced anisotropy is there in the C. elegans part. So these are the material parameter that we can extract from this analysis. And the thing is like that we can, so this is just technicality that we can construct with this intensity, velocity, and the width. We can construct a merit function which we will minimize and minimize with respect to these non-dimensional. Only alpha 1, alpha 2, alpha 3, these are the non-dimensional parameters of our model, and this is the turnover time scale. With respect to these four, we will minimize this merit function, and finally we can arrive on this the material parameters. So these are the values that now. So now at this point I can say that we can present you a quantitative measurement of the material parameters after analyzing those due to the laser ablation on actomyosin network. So in this sense, though we are applying this thing in two different organisms, but the process as a whole, if we can construct the theory well, then the process as a whole is kind of generic, and as the laser ablation is a very, very popular technique to probe the biological systems that we are, in this meeting, we are concerned about, particularly in the context of morphogenesis. So we can apply this process with the theory, and we can calculate the material parameters to understand the better. So now this is one way. So okay, so the talk is almost complete. So one way to estimate the material parameters. There is some the other way we also showed in our work that instead of analyzing the flow pattern or the flow behavior like the intensity analysis, the decay of the velocity, we can calculate or we can estimate the shape of the hole itself, only the hole, right? And why it is important, I will come to later. The shape, so these are blue points are shaped from the theory, and this is the shape from the experiment. So we can calculate the hole made by the laser ablation, the boundary of the hole. We can detect from the experiment, and the same thing we can detect from the theory itself, and then we can minimize the distance, the distance between the curves actually, and then finally we can minimize that distance with respect to the same our non-dimensional parameters, and it follows very. So this is our surprise. We exactly don't know why, but the thing is the parameters that this kind of minimization technique gives are the very close to the parameters that we got earlier. So why this is important? Because in many contexts, there is due to the geometrical constraints, we cannot measure the flow just after the cut in various biological contexts. For that case, we can, instead of that, if this is correct, and then we, instead of the analysis of the flow far from the cut, right, we can analyze the boundary only and get the physical parameters out of it. So here, actually, is almost done. So the point here, the conclusion is the thing that we are in the process of almost developing a method to estimate the material properties of cortex, or maybe in the tissue scale, or maybe in the, so these are the cellular scale experiments, but the similar process, similar theory can go into the tissue scale, and we can extract using the same like technique that we have a theory, we have the analysis from the experiment coupled together, and then we can calculate the important parameters that can control the flow or the pattern formation in tissue or in the cellular scale. So this is the, say, the conclusion of this talk. So